Thank you for this introduction, and it's a really great honor to give the annual Jack Elias Advances in Biomedical lecture. I still remember meeting Jack. I don't think he remembers when I was a postdoc at UCSF, and he came in and his talk basically blew me away. It was so brilliant, innovative, but the main thing was actually that really blew me away was his passion and his attention to people like me that really didn't count at this stage. So I asked some questions and he looked at me and basically gave the most excited explanation I could ever give. So this memory stayed with me. And naturally, when in 2012, sitting very, very comfortably in the jaw chair in Pittsburgh, Jack sort of very non-pompously said, would you be interested in a job at Yale? I definitely couldn't say, no, you're invited by one of the giants, you'd show up. And then I came in and it was love from first sight and I stayed and he left. So today I'll speak about pulmonary fibrosis. I think many of you know me. Feel free to also follow me on my Twitter kid, Kaminski Med. If you want to live tweet, please use the IPF Cell Atlas hashtag or the BlueUp4PF hashtag, which is the formal hashtag for the IPF Awareness Month. And then there's the other hashtags that I'm really usually tweeting about. The last one, ABCs, maybe the most unclear to all of you, but this is basically anything but COVID. And that's what I'm going to speak about today. I have many commercial interests. I'm consult multiple companies in the last 3 years. I have collaborations with Mergen, Versailles, AstraZeneca, Equity, and Pliant. I have an initiative in Vitix Therapeutics that I'm actually presenting at the Yale Office of Research, Blavatnik highlight today at 1 p.m. and many patents. But I wanted to start actually by speaking about something different and actually about the time of the year, September. Again, as somebody who grew up in a very hot country, a little bit dry, September living in the US is really exciting. It's the beginning of fall in Northern Hemisphere, the spring in the South, of course. It's the beginning of school year for children. As somebody who still have a high schooler, it's always exciting, although this year it's also a little bit anxious. It is also the renewal of academic year, and it's really great to see people coming in. The talks are coming, the conferences, everything is virtual now, but that's okay. Fellowship interviews, I always love to meet the future and the present of our discipline. It's so exciting. And of course, the Jewish holidays, Rosh Hashanah and Yom Kippur, not deeply religious, but this feeling of renewal, the fact that every person you meet, you say, may you have a good year, may your year be sweet and healthy, makes you feel good. And of course, Yom Kippur, the whole idea of atonement and forgiveness for the bad things you've done. In my family, though, September is not a good month. Because in September 1939 was the time that the childhood of both my father and my mother ended. My mother was 7 years old in first grade. My father was 13-year-old. And their childhood ended by mid-September 1939 with the beginning of World War II, with the invasion of Germans to Poland. And it was terrible. We only have, for my father, who was the sole survival of his family, we only have this photo remaining. It's his two sisters who both died. And in some ways, their causes of death have been driving forces in my career and my life. Because his sister Rachel died of pneumonia and poverty. And his younger sister, Hinda, died because of racism, ordinary evil committed by by ordinary people, and because of indifference. And for me, even as a 3-year-old kid, it was clear that I'm going to fight all my life disease, poverty, and racism. I do want to be a little bit more on the positive side. And this is a photo of my mother. Here you can see it In 1946, a year after the world ended, she was 14 at the time, had 2 months of school between the age of 7 and 1946, never went back to school, worked all her life. She still is doing things. And her smile at that age, which hasn't changed, is for me really important because there is a recognition of privilege in the good way. Being happy that you're healthy, that you don't suffer pain, that you can afford to have food for yourself and for your kid and for your safe is a big thing for my mother. And for me, the recognition of privilege and also the recognition of solidarity with the sick, the poor, the dispossessed, the disadvantaged, the excluded, the harassed, the oppressed is personal. And it's really derived from this look at my mother's smile after the war, the hope of a person who actually at that time didn't have anything. She built a life and educated as a single mother, relatively accomplished kids. But the main thing that I cannot let go is even now, the risk of dispossession, of disadvantage, of poverty, of exclusion, of harassment, of oppression, of racism is real in our life, in our world, in our society. September is also a month, there's actually, I counted, there's around 20 awareness months in September. And the closest to my heart are two. One is Pulmonary Fibrosis Awareness Month. As you can see here is on the left, you can see a few years ago when I came to Yale, there was a patient, Diane Reichert, she had pulmonary fibrosis, and she started this event of dye your hair blue or blue up for PF, for disease awareness, because her experience was extremely frustrated. Nobody knew what is this disease. I was following her campaign, but it didn't really gain a lot of traction. So we decided to make an event in this section to show our solidarity. And it was really, our offices became a hair salon. We all congregated, dyed our hair blue, made a video. You can find it at YouTube. But for me, the look on Diane's face, the feeling of recognition, the happiness, despite her disease, was worth the whole day and probably many more awards in my career. Diane passed away 6 months later, but the campaign picked up and the Pulmonary Fibrosis Foundation every year uses her slogan. So this is a driving force. September also has been declared the Women in Medicine Awareness, Women in Medicine Month to advance equity, drive change by the American Medical Association. Most recently, there's been the campaign this year of Her Time Is Now, and I was very proud to be invited to participate in the first flash mob. But I feel very strongly about a very simple idea, which is an idea that I was not completely aware of until a few years ago, which was that many of the people that I really deeply respected in my profession, amazing, talented physicians and scientists, never could express their talents in fully because of one reason. They were women. Actually, because of one more reason, because there were men around. And when this awareness sort of crystallized, there was no way back. I became very active, and I see the role of men as allies in the campaign for gender equality, for ending harassment is critical, and really hope that this is something that we can finish in our lifetime. And of course, I added sort of a small photo of myself kneeling at the exciting day in, I think, April or May, that we all congregated outside of Sterling Hall of Medicine for the Black Lives Matter events of the medical school. Again, it was amazing to see the leadership of early career physicians and scientists taking over, expressing a commitment to change the evils of hundreds and hundreds of years. So for me, patients with idiopathic pulmonary fibrosis are a little bit the unseen or the disadvantaged. And from now on, for the rest of my talk will be about idiopathic pulmonary fibrosis. It's a chronic progressive form of fibrotic interstitial lung disease. It's defined by exclusion of known causes of lung disease. There's around 200,000 patients in the US, 6 million worldwide. It has a very typical pattern. I'll mention it in the end. The facts to remember is that the incidence increases with age. So at certain ages, it's actually more common than most of the diseases you have heard and cared about. It is extremely rare below 55. The median survival is 3 to 5 years, but the range is high. And my group, thinking about this idea that when you meet a patient and you make the diagnosis of IPF and you speak with them about the prognosis, and then go back home and their kids do a Google search, they found this death sentence. You're going to be dead. 50% are dead within 3 to 5 years. But actually, there is a big range. And some patients need care earlier. Some patients can be stable for a long time. And part of my work has been basically trying to identify biomarkers. And I'm glad to say we found them. I will not speak about it today. There are 30,000 deaths a year in the US. 1% of the deaths on death certificates before COVID-19 in the UK was pulmonary fibrosis. The only curative treatment is lung transplantation, and there's a strong genetic component. This is the typical lesion of the fibrotic lung, the usual interstitial pneumonia pattern on histology. We usually see fibrosis and remodeling, frequent honeycombing. The main thing is that the involvement has to be peripheral, and has to be a mix of normal lung and involved lung. And we usually see the lesion of IPF, fibroblastic foci at the edge of dense scars with sort of epithelial cell lining them. And this will come in afterwards. And of course, we need the absence of indicators of other lung diseases. In recent years, we do less and less biopsies because actually the usual interstitial pneumonia pattern on imaging is actually relatively stereotypic. It should be peripheral predominant. You can see the CT scan on the left, basilar predominant, reticulation, honeycombing, again, an absence of atypical findings like ground glass opacities, mosaic continuation, gas trapping, peribronchial distribution, or other consolidation of micronodules. So peripheral predominant, basilar predominant, reticulation, and honeycombing, traction, bronchiectasis, these are the things you want to look for. And of course, the diagnosis, because it's a relatively rare disease and the presentation is very non-specific, basically dry cough and shortness of breath, requires a multidisciplinary group. And at Yale, we've been fortunate to have master clinicians like Danielle Antinosarkis and Mridu Gulati and Jonathan Seiner, outstanding chest radiologists like Amy, and pathologists like Rob Omer, and sort of superb translational scientists like Erika Herzog. So it's a really exciting group, and the multidisciplinary discussions are usually very, very lively and informative. When I started getting interested in pulmonary fibrosis, I finished my fellowship in 2000. The paradigms of pulmonary fibrosis was relatively simple. It's a stable, invariably progressive disease. There's some kind of an inflammation that leads to fibrosis. This was mostly derived from animal studies from mice. And there was sort of a very passive view of a disease of fibroblast proliferation in response to inflammation with extracellular matrix deposition and decrease in extracellular matrix degradation. And the notion was that it was a boring disease. The lung is just a fibrotic sac, and you can never reverse fibrosis, just need to suppress inflammation. In fact, I was told by one of the editors of the most important textbook in pulmonary medicine that if I go and do pulmonary fibrosis, I will not have a career. The beginning of my career, I was there when the gene expression studies and the ability of measuring initially 100 genes, then 500, then 1,000 genes, then the whole genome became available. And my second research, after failing miserably in my first research projects, I started working on gene expression microarrays at the real beginning. And one of the first thing I did, coming from, again, from a deep clinical background, was to obtain human lungs with pulmonary fibrosis through a collaboration with Roche and Stanford and analyze them. And basically, this analysis, which we did 6 lungs or 8 lungs, identified themes that we kept identifying afterwards, and in some ways was really innovative because it showed that in human IPF lung, it wasn't just dead tissue. There was an active network of gene expression changes that suggested an active process of remodeling. Over my career, we sort of developed it, and from 6 lungs, we went into 571 lungs. Here, what you see is a heat map, a common way that we show gene expression data because it's really hard to grasp. Every column is a patient. Every row is a gene. These are controls, COPD, IPF, other disease. Yellow is increased, and purple is decreased, and gray is unchanged. And you don't need to be a bioinformatician to see that the IPF lungs are very different from COPD or control or other disease. And we've identified probably 2,000 genes that are differentially expressed. Now, this is in a very simple way. We take the lung or lung biopsy, we grind it, and we check all the RNAs in it. And we found around 20 pathways and functional themes that distinguish IPF from controls, including this active degradation of the lung, the activation of developmental pathways, mostly the Wnt and beta-catenin pathways, evidence for TGF-beta downstream effect, a shift in epithelial cell phenotype, and enrichment for microRNAs and other things. And this has led to big projects in my lab of trying to find microRNA therapeutics in pulmonary fibrosis. And this was neatly summarized by Milica Vukmirovic in my lab a couple of years ago. And the other thing that we've learned about IPF is that it's really strongly associated with aging. You can see here that the numbers per 100,000, and basically in men, when you get over the age of 80, it's almost 500 per, over a 500 per 100,000. It's really very common in the elderly. We also learned that the disease has a strong genetic background. So 5 to 20% of all cases have a family history of pulmonary fibrosis. Sporadic disease is associated mostly with SNPs in the MAC5B promoter, but also telomerase and other genes. So 30 to 50% of sporadic cases are related to genetic variants. And there's also specific mutations that cause hereditary pulmonary fibrosis. In 2014, we were really excited because actually 2 drugs were approved on the same day as the first-in-class antifibrotics, nintedanib and pirfenidone. Both reduce disease progression in IPF by 40%. They have similar effects on other progressive fibrotic lung diseases, may have effects on mortality. They have no effect on quality of life, so the patients don't really feel they benefit. We also learned that immunosuppressive increased mortality. And now we have many, many clinical trials to follow up on them. We also have sort of a new set of paradigms. And again, this is 2020. So the same thing we observed in 2002, central role for shift in epithelial cell phenotypes, the activation of remodeling networks, including matrix metalloproteases and aberrant extracellular matrix. We have identified that myofibroblasts are active cells, not necessarily the sort of the passive cell responding to inflammation. Strong genetic component, as I say. That the association with aging and the genetic association with telomere mutation has led to identifying the role of telomere attrition and mitochondria in the disease. Developmental, the role of Wnt signaling, microRNAs, TGF-beta, and ER stress. And we also have the sort of, we know that immunosuppression kills the patient, and we know that some drugs slow down the disease. The other thing, and again, mostly because there's a early career and residence in the crowd, this is something when you pick a career, you want to know which field is really exciting and booming. So here is a diagram of all papers in PubMed, normalized to 2002. And the dashed black line is all the papers in PubMed. And as you see, there's actually number of papers has increased by nearly double from 2002 to 2019. And of course, this tracks cancer literature because the most papers you have in cancer literature. But if you look at other diseases, many of them have actually been below the publication rate. But look at IPF, look at this red line. This is the number of papers related to IPF. Our community has nearly gone tenfold in the number of publications per year from 2000. This is the most booming and exciting field. You know, we're really like where cardiology was in 1990. Our biggest drugs are coming in. Our biggest discovery are coming in. Much of it is driven by genetics, by new drugs approved, by genomics. And I hope that some of the things I'll show you today may drive the next boom. So what do you think we think happened in IPF? So again, this is the typical lesion of the idiopathic pulmonary fibrosis lung. You have normal looking lung adjacent to the typical lesion of the IPF lung with sort of the myofibroblast foci and this thin linear thin layer of epithelial cells that we used to call modified type 2 epithelial cells or other. We had different names from them. And what we think is happening, and the main purpose of this image is, of course, to show you that I can make really cool GIFs. And also that actually this was created initially by Arnaud Maillet here at the Yale. And basically what it means that we have recurring microinjuries and epithelial cell injury that repeat. And then we have cells that are dying. Others get activated, epithelials, and they activate fibroblasts and macrophages. We start depositing extracellular matrix. This changes the stiffness of the area. And now we have cell proliferation, accumulation, and migration, which causes the lesion of usual interstitial pneumonia, especially if you have genetic predisposition, ongoing exposure that is not dramatic, that does not kill you, and telomere attrition. So some kind of a desynchronized aging. The only thing is that this model is really, really appealing, but it's actually not very specific. It's not telling us when I say fibroblasts or epithelial cell, what do I actually mean? And when I speak about pathways, when I say apoptosis, what does it actually really mean in reality? So our group has done a lot of work, but what I chose today in the next 20 minutes is to tell you about one effort we've been involved in and actually led. It's the IPF Cell Atlas. In the last 3 years or 4 years, we have had a major technological breakthrough that's going to affect our understanding of every disease, which is we have now the ability not only to profile an organ or a region, but we can actually profile the gene expression and potentially protein expression of every single cell. So in the IPF community, there's been a multi-center grassroots effort to create a comprehensive map of all human cells in the lung of patients with IPF and other fibrosis interstitial lung disease. Of course, there's an effort to create also a reference, the Human Lung Cell Atlas. And the idea is to accelerate novel, accurate, and effective tools for diagnosing, monitoring, treating disease, and to create a simple to use interface to allow exploration. Now, this is the image you're looking, sort of this three-dimensional image that you're seeing, is actually the data. So the data is huge, and actually every little spot here, every little dot, there's 300,000 cells in this. Every little dot is one cell, and this is colored by the cell types and basically clustered the way these cells are organized in relation to each other. So really hard to see the data. But what we created is basically the IPF Cell Atlas. If we were now at Fitkin, I would ask you all to raise your iPhones, scan, turn your camera at the QR code. And basically, if you did that, you would gain access to the IPF Cell Atlas, in which really you could search for every gene you're interested in, every cell you're interested in, and see what they do, how they change in disease or in health. So please feel free to do it. The website is www.ipfcellatlas.com. And this is sort of the data sets that are available. There's sort of 4 groups that contributed. I'll mostly focus on our efforts that have been published in Science Advanced this year by this. It's a mega amazing work by Taylor Adams, Jonas Schupp, Sitin Yang, Farida Angari, and Nir Newmark that actually created the website, and in tight collaboration with Ivan Rosas's group. We profiled samples from 78 human lungs, 32 IPF, 28 controls, 18. We profiled 312,000 cells. So what did we learn? The first thing we learned, and what we do is when we look at the data, so we look at the cells and we assign them roles, and we can identify different things. And again, as I said, it's really impossible to look at all of the cell populations at one time. So when we analyzed the immune populations in the IPF lung, we found that the cell that really was dominant was the macrophage. And actually a macrophage that looked like a profibrotic macrophage. And this is the image of all the immune cells in the lung. As you can see, again, every dot here is a cell. And what we did, we colored the dot based on where they come from, which disease. So red is IPF, yellow is COPD, and blue is control. And as you can see, even sitting at your computer or your iPhone back home, or on the floor, or wherever you are, you can see that for most of the population, there's no big difference. However, there's this really big red blob, which are macrophages. And these are very unique macrophages. They express a set of markers. CHI3L1, YKL40, which has been discovered by Jack here and emerging as a key regulator of disease. MMP9, phospholipase A2, osteopontin that was first studied actually by Jeff Chubb, the director of our asthma program when he was a fellow at BU, and many other things. And what's interesting about these macrophages is that one is they actually show up in other data sets. So this is not like something we discovered for new, but in 3 of the 4 data sets, these macrophages dominate. And people have done animal models of disease and show that these macrophages actually have an active profibrotic role. The other thing, it's always tempting to think that things are similar. And what I'm showing you here is basically a 3-dimensional image of the lung macrophages. And I'm coloring them based on the different markers. Remember, I said that they have osteopontin and YKL40 and MMP9. And as you can see, I hope you can see, not all the cells express all 4 markers. So again, there may be different levels of differentiation that we could still define. But when we classify the macrophages, and basically this is a heat map of genes, and basically what we did is we took the macrophages and went from non-fibrotic to fibrotic, we can actually identify a signature of these profibrotic macrophages. And you can see it highlighted in the pinkish diagram. And here's the thing, what I find really exciting about that now we don't have to target one molecule. What we need to show is that you can normalize a macrophage. So we take macrophages from the IPF lung, we can treat them with multiple therapies. And for instance, and basically see if we normalize them, which again may allow us to generate much better therapies than what we had before. The second insight is that epithelial cell populations are markedly shifted in the fibrotic lung. And there are some, we actually discovered novel cell types. So remember, the structure of alveolus is very stereotypic. There's alveolar type 1 cell, alveolar type 2 cell, endothelial capillary, an alveolar macrophage, and even sometimes a fibroblast. That's it. There shouldn't be any other cell type there. And when we look at the IPF lung, and here is, you can see basically just a % makeup of epithelial cells in the lung parenchyma. The first thing you see, and we color this again, IPF is red, COPD is yellow, control is blue. We keep the same color scheme. And you see that in the IPF lung, type 1 epithelial cells, the cells that do gas exchange, are really decreased. And so do type 2, which are the cells that secrete surfactant and serve as progenitors for the type 1 cells and are also responsible for the fluid balance in the alveolus. On the other hand, ciliated cells, airway cells, are really increased. And this is a dramatic shift. Basically, it suggests that the lung parenchyma undergoes, the distal lung undergoes proximalization. Airway cells are moving or are increasing within the lung parenchyma. And naturally, if you think about it, if you lose your type 1 cells, and that is designed for gas exchange, and you replace it with the ciliate cells that were designed for actually mucosal immunity, you're not going to be able to breathe. And this was found also in other studies. So we were able, when we looked at epithelial cells, now we can classify them based on known markers. And we found all of the known epithelial cells in the human lung. But we also found a group of cells that was very different, and you can see them highlighted in the Yale here. So these are all the epithelial cells in the human lung, colored by disease. Red is IPF, yellow CPD, blue is control. And you can see there's this group of epithelial cells that are very different from everything else. And what are these cells? They're really strange. They have a set of markers that really suggest that they're sort of out there. They carry markers of airway basal cells, especially P63 and keratin 17. Airway basal cells are sort of proximal lung cells that serve as local progenitors. However, they're not completely basal cells. That's why we call them basaloids. They are epithelial cells. They express epithelial markers, but they express also mesenchymal cells, fibronectin, collagen 1, and cadherin, HMGA2. So they're cells that have a component of epithelial to mesenchymal transition. They have a machinery to activate TGF-beta, especially the integrin alpha V beta 6 that has been a therapeutic target. They have markers of senescence. They have a transcriptional program of stemness, especially SOX9 and SOX11. And they carry some of the most known biomarkers for IPF, such as MMP7. And this is just a stain because we wanted to know where these cells are. And when Jonas showed me these slides, I was really impressed because these cells are really sit at the edge. Remember, I told you the myofibroblast foci is the lesion of the disease. And these aberrant basaloid cells sit at the epithelial, at the edge of the myofibroblast foci. So think about it. This is a cell that has all the machinery to drive fibrosis. It's sort of strange and really seems to be sitting at where the action is happening. And this is just a stain from the paper from Nick Banovich and John Kropsky showing the same results, again, showing that these are not results that show up only at Yale and Baylor. They also happen were verified by multiple other scientific groups. So the question, of course, as a physician, I'm always interested in curing stuff, right? So should we target aberrant basaloid cells? Can we follow the markers? So I won't go into detail, but we think that we can. And I'll mention one of my things. Our group actually has discovered many years ago that MMP7, matrix metalloprotease 7 or matrilisin, is probably a really good predictor of outcome in the patients with IPF. If you measure it in the blood, if it's high, the patient has worse prognosis. If it's low, the patients are stable. This protein doesn't change. So if you have high levels, it doesn't go down. And most of our current therapies don't change its levels. So the question, if you have a therapy that change MMP7, and you're targeting airway aberrant basal cells, and MMP7 changes, maybe you know you hit them. So this is important. Insight 3, which is really, we feel is sort of another show of the power. When you do unbiased analysis, you see things you don't necessarily care about. And again, I study mostly epithelial cells, never pay attention that much to endothelial cells, except for my PH team shows up in my office. But when we looked at endothelial cells in the IPF lung, we actually discovered, and you can see here, again, remember the same color scale, red is IPF, yellow CPD, blue is control. And we discovered basically there's a whole population of endothelial cells that was not described before that is really increased in the IPF lung. they have a set of unique markers, and the most important is collagen 15. And we call them basically peribronchial endothelial cells, because in the normal lung, they never show up in the lung parenchyma. They're never in the endothelium. But in the, they only show up in peribronchial regions, and also pleural adventitia. And Jonas Schupp is writing now the Endothelial Cell Atlas to describe them, together with Rob Homer and Taylor Adams. But in the IPF lung, they're really amplified, and you can see them in the States. And again, think about it. If your lung has been evolved for gas exchange, and it has a set of endothelial cells that are really adapted for gas exchange, and now you have a population of endothelial cells that is not of the pulmonary circulation, that's probably systemic circulation, that's sort of migrating and enriched. Again, your ability to breathe is going to be reduced. Are these cells targets? I don't know, maybe. I think it's very interesting, but the main thing is actually that this shows us again this sort of abnormal replacement of normal distal lung with proximal lung cells. The last insight again is sort of, and there's many more, but is about fibroblasts. And basically, there's the famous sentence, what's in the name, which we call a rose by any other name should smell as sweet, from Romeo and Juliet. And Juliet was right. There's more to fibroblasts and myofibroblasts than the name. When I speak about myofibroblasts foci, doesn't really mean that I know what they're doing. And when we look at fibroblasts and myofibroblasts in the IPF lung, and I'll go very quickly about this, we basically see something very simple. Although we tend to assume that the myofibroblast comes from a fibroblast and a fibroblast becomes a myofibroblast, that's not what we really see. The subpopulation of fibroblasts and myofibroblasts tend to connect within the cell types and not within the disease states. Just to simplify it, we can take all the fibroblast-like cells in the lung and cluster them, and what you get is this beautiful butterfly. And again, I color it based on disease. Red is IPF, yellow is COPD, and blue is in control. And basically what you see is that the myofibroblasts and the fibroblasts are completely distinct. And if we actually do a pseudo-time analysis, so go from where is fibrosis highest to where is fibrosis lowest, we see two trajectories, but going from no fibrosis to fibrosis, but these trajectories are within cell types. So I will not belabor the facts, but I'll say that this analysis, which is called archetype analysis, suggests that we may be dealing with disease-related fibroblasts and myofibroblasts that actually originated independently. They have different markers, and maybe we should actually target them differently. They actually have different markers. Of course, there's the philosophical question, are these the right names? Does it really matter? So how do we actually handle this? How do we start moving to cure IPF? So I've shown you an amazing array of anomalies. And when we do a sort of a network analysis, and this beautiful work and figure was done by Taylor Adams. If we look at this is a gene cell regulatory network in the control lung. Every color is sort of a cell line, and you can see the domain of influence of the different cell types. And this is IPF, and you can see that the control lung is very, very balanced. The IPF lung, on the other one, is really fragmented and is really dominated by certain cell types, the aberrant basaloid cells, some of the fibroblasts and macrophages, as we predicted. But there's also a really wide range of anomalies. So one of the things that we ask ourselves, can we normalize a cell type? And we use the MIT generated connectivity map. It's a library of cell culture experiments in which cells were stimulated by thousands and thousands of compounds, and you could sort of go there. You could take your cellular gene expression and see if you can reverse it. And we were able to identify a set of compounds that will reverse IPF aberrant basaloid cell phenotypes. and we found a set of compounds that will reverse IPF myofibroblast fibroblast phenotypes. Some of them overlap, but the majority don't. But the thing is that this data set was not generated for fibrosis. So ideally for us, we would like to actually do this with human lung and explants and basically treat them with thousands of drugs that have already been in humans and we know that they're safe, and see if we can really normalize the lung, because this would be the quickest route to #cure IPF. So can we cure IPF? I think we can. I think the extent and dynamic range of the changes probably suggests that there's a lot of things we can reverse. Does it mean, and when you think about it, this is the new model. Remember, I showed you the GIF at the beginning. This is the new model. This is sort of a diagram, again, done by Arno, of the normal lung, the distal lung with the ciliated basal cell club cells, and then sort of the alveolus with the macrophage, type 1, type 2, and endothelial cells and fibroblasts. And basically, after injury, you get type 2 cell proliferation, but because of genetic predisposition, ER stress, short telomeres, these cells cannot really replicate. They cannot compensate for the type 1 death. So what we get is secretion of dumps, mitochondrial DNA release, as has been shown by Wonnie Ru and Erika Herzog, is important. And then activation of myofibroblasts and macrophages. And basically we get this stiff matrix secreting of more dumps, recruitment of circulating monocytes, and at this stage, really hard to get in. The other thing is the lung starts sensing that something has gone wrong, that there is a local failure of progenitor cells, and then you get recruitment or increase in aberrant basaloid cells, an activation of an airway building program that leads to this proximalization of the distal lung. And basically this rebuilding of the lung without a blueprint is what causes fibrosis. So the neat thing about this is, of course, a horrifying model. But the neat thing is that there's multiple places we could intervene. And we have the knowledge. And now we have our sets of targets. So I believe that together, all of these things can lead to the road to cure IPF. So our key discoveries, as I've mentioned, the aberrant basaloid cells, the collagen 15 endothelial cells, profibrotic macrophages, the unique features of myofibroblasts and invasive fibroblasts, the phenotype of all the population, and this very impressive phenomenon of proximalization of the distal lung. And then we have targetable frameworks. We know now that we need to target more than one cell. We can identify now the sort of active networks. And we know now that probably one way forward is to generate this IPF connectivity map. So again, I invite you all, go to the IPF Cell Atlas, find your target. It's a lot of fun. So during the talk, I mentioned the people that whose work was most relevant. But again, I want to highlight sort of the disease awareness and the connection with the patients. Our missions are really to do outstanding clinical work, to provide outstanding education, to do cutting edge research. But when you think about it, there's a unifying line to it, which is it's all about the patient. And this is, again, photos from this IPF awareness event that we did a few years ago with Diane Reichert. It was a lot of fun for us. It was really important to the patient community. So this talk is dedicated to all PF warriors on PF Awareness Month. And it's also really dedicated to the amazing teams of Yale Pulmonary Critical Care and Sleep Medicine. In the 7 years I've been here, I've always had the feeling that I'm working with a unique group of individuals and also a superb team. And during, of course, the COVID-19 surge, my intuition was completely fulfilled. So Yale PCCSM, there are COVID and everyday heroes, and thank you. So I'll stop here and take any questions you may have.
Thank you very much, Naftali, for a really wonderful talk.